right, so we can get started. So welcome, everyone. I'm Megan. I'll be moderating today's Open Box Science Genomics seminar. So we have two exciting talks lined up today, which complement each other very well. They're both centered around leveraging deep mutational scanning data. So our first speaker is going to be Dr. Mireia Seuma from the Institute of Bioengineering of Catalunya in Barcelona. Her talk is titled An Atlas of Amyloid Aggregation, the Impact of Mutations on Amyloid Beta Fibril Nucleation. And then our second speaker is going to be Dr. Amar Tareen, who is currently employed at Regeneron, formerly the Cold Spring Harbor Laboratory. And his talk is titled MAVE-NN, Learning Genotype Phenotype Maps from Multiplex Assays of Variant Effect. So at the end of each talk, we'll have some time for questions. So feel free to enter your questions down in the chat or ask them here on Zoom. So we can get started. So it's my pleasure to first introduce Dr. Mireia Seuma. So she is currently a postdoctoral researcher in Benedetta Bolognesi's lab at the Institute for Bioengineering of Catalunya in Barcelona. Dr. Seuma uses deep mutational scanning to understand and predict how genetic variation impacts amyloid fibril formation in Alzheimer's and other neurodegenerative diseases. So with that, I'll hand the floor over to Mireia so she can begin her talk.
Can you see my screen?
Yes.
I'll make this full screen. I'll add the pointer. Right, so you should see this now. Great. Hi, everybody, and thanks, Megan, for this introduction. I'm happy to be here today and talk about my project in which we mapped amyloid nucleation by the mutational scanning. I did my PhD at Benedetta Bolognesi's lab at the Institute for Bioengineering of Catalonia. And before starting, just a big thank you to people in my lab, especially Benedetta, and also our collaborators at the CRG, Ben and Andre at Ben Lehner's lab. And so in our lab, we are interested in proteins that self-assemble, and we have a special focus on amyloids. Amyloids are these huge and beautiful structures. Here, what you are seeing, this is an amyloid fibril, and they are composed by identical monomeric subunits that, in this case, this is adopting an S shape, and many of these monomers stack one on top of the other to form a fibril. And there are many proteins that can form these amyloid structures, and here you see different examples of different proteins, and now we have structures that have been solved at very high resolution by a traditional structural methods such as cryo-EM or NMR. And you might be also familiar with amyloids because when these things deposit, they are associated with many human diseases, especially to neurodegenerative conditions such as Parkinson's or Alzheimer's. And we have an evidence that there's a causative link between these proteins that form amyloids and the disease, because we know from genetics that one single mutation is enough to make your brain shrink, like this example here. And once these things form, they are really like rocks. It's very difficult to disaggregate them. And in fact, amyloids are thermodynamically the most stable states in the free energy surface. But thankfully, this doesn't happen all the time. And this is because the process by which they form is controlled by nucleation. This means that the monomeric subunits need to form a first critical nucleus and assemble that require a very high kinetic barrier. Then they can grow and seed further aggregation into amyloid fibrils. And nucleation is the critical step in here because it requires this high energetic contribution, and it's also an irreversible process, meaning that once it starts, there is no way back. And we have all those structures solved at high resolution, but we still don't fully understand the mechanism by which they form. And this is because we don't fully understand nucleation. And to further characterize this process, we took as a classic model of amyloids, the amyloid beta 42 peptide. This is the main component of amyloid deposits in the brain of Alzheimer's disease patients. And this is a cleavage product from the amyloid precursor protein, which, depending on the cleavage site, the resulting peptide has a different length. So in our body, we have different isoforms, and one of the most famous ones is a beta 42, which is the one that here we are taking as our wild type. This has 42 residues, and the last 14 residues, all of them are glycines and hydrophobic amino acids. And just for simplicity, I will call this part of the peptide the C-terminus, while the N-terminus, which is the first part of the peptide, is full of positive, negative charges and also polar amino acids. And now we have different structures that have been solved for A beta, different structural models, but I'm particularly interested in two of them because these were solved very recently from samples that were coming directly from patients with Alzheimer's disease. And these were solved by cryo-EM. And these two structures, they have a very similar monomeric subunit with a very similar shape, in which the C-terminus, those hydrophobic residues, are forming the amyloid core of the fibrils. And instead, the N-terminus has a bit of a different orientation, and it remains disordered in most of these structures. A beta is also a very interesting peptide for us because mutations in this peptide have been are known to cause familiar forms of Alzheimer's disease. In this matrix, what I'm showing you on the x-axis is the wild-type amino acid at each position, and on the y-axis, all the mutant amino acids that we can introduce. The black squares indicates the location of the known mutations that are associated to familiar Alzheimer's disease. And there has been quite a lot of work, especially in vitro, to understand the impact on aggregation of these mutations, and the ones that you see here in blue are known to increase aggregation, the ones in red are known to decrease aggregation, but there is no consensus on the effect of the disease variants, what they do in terms of how they impact amyloid aggregation. And there's not even one study in which all of these mutations have been tested together. This is also a very empty matrix, meaning that for the rest of the mutations, we have no information. We are not able to predict whether they can be pathogenic or not if we ever find them in a patient. So we are currently classifying them as variants of unknown clinical significance. And if we think about population size and mutation rate, all of them are very likely to exist in at least one person living in the planet today. So here we asked ourselves, do we have a way to test all possible mutations and not only the ones that are associated to disease? How do they impact amyloid nucleation? And to do so, we are using a technique which is called deep mutational scanning, which allows us to test thousands of sequences in parallel in one single experiment. So the idea here is that we build a library of variants. In this case, this was a library of variants of A beta that we introduced in a model system. This was done in yeast cells, and here all variants are represented equally in the population. But then we introduce a selection step. So now we are going to discriminate the variants in the population for a specific phenotype. And here you can imagine any type of selection. One of the simplest examples to think about is toxicity. So if there's a variant in the population that is toxic, this will be depleted after a certain number of generations. But if instead there's a variant that is not toxic, but it's neutral or even beneficial, this will get enriched in the population after a certain number of generations. And so we can literally quantify the performance of each variant by sequencing the population before and after selection and just compare the presence of each variant. And as I was mentioning, DMS allow us to test many sequences in parallel in one single experiment. I applied this to amyloid nucleation, and this is how our selection assay works. Let's now forget about libraries for a second, and let's just imagine that we have A beta 42 wild type. we fuse this protein to the nucleation domain of sub35. Sub35 is a well-known cis prion and a translation termination factor. So when this remains soluble, this is functional, and its job is to recognize premature stop codons. So here we are using a GIS strain which has a premature stop codon in the adenine gene. This means that when it's active, sub35 is recognizing the stop codon and the cell is not producing adenine. So if we don't provide adenine in the medium, this cell cannot grow. But now let's move to the second scenario in which this A beta starts to nucleate. It will cause the nucleation of sub35 and recruit full-length endogenous sub35. This sub35 is not doing its job anymore, it's not recognizing the stop codon, and here the cell starts to produce adenine. So here the cell is growing in a medium without adenine. Everything you have to remember from this selection assay is that we have a way to relate amyloid nucleation to cell survival in the absence of adenine. And if we now introduce a full library of variants, we can perform the same experiment and deep sequence the population before and after selection and calculate what we call a nucleation score. And why do we call them nucleation scores? Well, because the scores that come from the DMS experiment correlate very well with in vitro kinetic rate constants of amyloid nucleation. Let me show you the power of this type of assay. So we were in front of this empty heat map, and we could fill it in with information for virtually all single amino acid substitutions on their effect on amyloid nucleation. What you are seeing here in this matrix, again, on the x-axis is the wild-type amino acid at each position, and on the y-axis, the mutant amino acids that we are introducing. The color code indicates you the nucleation score, so in blue means increased nucleation, and in red means decreased nucleation, always relative to A beta 42, which is our wild-type. One of the first things that we asked ourselves here is how do the disease mutations score in this assay, and you can see them here again in a black square, and all of them have a positive nucleation score, meaning that all of them increase nucleation. We also tested the performance of the nucleation scores in discriminating disease variants, and the rock curve here in blue shows you that nucleation scores accurately discriminate all of them, and they do it better than any other computational predictor or experimental assay done before. So ultimately, human genetics is proving that this assay is relevant to familiar Alzheimer's disease. If we now go back to the data, one of the most striking things here is that mutations have a very different impact at the C-terminus compared to the N-terminus. And we are using the structures to rationalize the impact in the different positions. So here on top, what you are seeing is the percentage of mutations that increase nucleation at each position, and below the percentage of mutations that decrease nucleation. You can see that the ones that decrease nucleation are highly enriched at the C-terminus, while at the N-terminus, mutations have a bit of a more balanced effect. And there are many other things that we can explain about this data. We could spend hours just describing interesting things here and there, and I'm just going to point out a couple of them. So for example, if you checked what happens when we substitute two positive charges, they can increase nucleation, but this is a very different scenario from substituting two negative charges. Or for example, introducing aliphatics, they are not tolerated at the first 10 residues of the C-terminus, but from residue 12 onwards, they can increase nucleation. And this impact of hydrophobic residues is something that current predictors would have never guessed. And in fact, we try to correlate our data with all predictors that are available out there for aggregation, solubility, deleteriousness, anything that you can imagine, and none of them was able to recapitulate the effect of mutations on nucleation. And here we think that we really need to perform this type of high throughput experiments if we want to understand what drives amyloid nucleation. And once we were finishing this project, we started to be very interested in other type of genetic variants, such as insertions, deletions, large deletions, or even truncations. And this is because, well, they exist and they cause human polymorphism. They cause disease and they are associated to disease, and they are useful perturbations, meaning that they ultimately allow us to explore larger sequences space. And does this matter for A beta? Well, yes, because some of these truncations have also been found in pathogenic amyloid deposits and are known to increase aggregation. And here I'm showing you an example. This is a classic in vitro kinetic studies in which aggregation is followed by means of thioflavin T binding. And there's at least one truncation for A beta 42 that increases aggregation. And the good news was that in our assay, we can throw in any type of variants that we want to test. And so this is what we did. We built a library that it contained not only substitutions, but also insertions, deletions, and large deletions and truncations. These are the distributions of the nucleation scores for each of these subsets. And although there's a large fraction of variants for all of them that decrease nucleation, that have a negative score, there are also quite a few variants for all data sets that can still increase nucleation. And let's go through each of these subsets. Let's start with single amino acid insertions. This is the heat map of insertions. In this case, I'm adding one extra amino acid at every position at a time. And this heat map is quite similar to that of substitutions, but it has some differences. And for example, it allows us to identify a specific stretch at the C-terminus that is between residues 33 and 38, that virtually none of the insertions is tolerated. And here we think that these residues constitute an inner core of the nucleating sequences that cannot accommodate additional side chains. Here, substitutions would be allowed, some of them, but not adding one extra amino acid. Let's now move to single amino acid deletions. In this case, I'm removing one position at a time. And again, we see that deleting positions at the N-terminus can increase nucleation, but we cannot remove one single amino acid at the C-terminus. And the top scorer here is the deletion at position 22, which is the only single amino acid deletion known to be associated to disease. We can delete bigger chunks of the peptide, and these are truncations from the N or the C-terminus. And let's start from deleting residues from the C-terminus. This is a bit of a boring plot. None of these truncations is able to increase nucleation, but I'm just showing you this here for sake of pointing at this variant. This is a beta 40. And as I was mentioning at the beginning, we have this isoform also in our body, and it's known to be less aggregation prone than a beta 42. So it's good to check that here is behaving as we were expecting. If we truncate the peptide from the N-terminus, now we see that we can increase nucleation by deleting it from the N-terminus as long as we don't enter the C-terminus. And the top scorer here is this variant. We call this the K core because it's literally a K, a lysine, just in front of the amyloid core. And this is sufficient to boost nucleation. We also tested this individually and confirmed that one single positive charge in front of the core aggregates faster than the wild type. And not only the lysine, which would be the wild type amino acid, but also an arginine just in front of the core would behave the same. But this is not the case for negative charges in front of the core. Let's now finally move to large deletions. So these are we are now deleting chunks of the peptide in different sliding windows inside the sequence. This is a summary of all these deletions, and on the y-axis, on the x-axis, you are seeing the first deletion, the position that I'm deleting, and on the y-axis, the last position that I'm deleting. And just to give you an example, if we take this little square in here, this would be a deletion that starts at position 26 and goes until position 38. If this is the wild type sequence, this is how it looks upon deletion. And we can also divide this data between those deletions affecting the N-terminus, deletions affecting the C-terminus, or deletions affecting both the N and the C-terminus. And we see this trend of mutations highly impacting the C-terminus, but increasing nucleation at the N-terminus. And at the N-terminus, we could identify a specific area in which many mutations increase nucleation. We define this as the hotspot of deletion of X, and these are all deletions that remove residues within 17 and 27. And after some time that we had generated this data, there was this paper that came out, and it was describing for the first time that there's a large internal deletion in the A-beta peptide that is associated to disease. This is known as the Uppsala mutation because it was found in a Swedish family. It's a deletion from positions 19 to 24. And so we went back to our matrix and checked the nucleation score, and we confirmed that it also increases nucleation here. And in fact, it lies inside the hotspot of deletion effects. But what is a bit alarming here is that this mutation is surrounded by many other mutations that are very similar to this one that also increase nucleation and are currently of unknown clinical significance. So we think that we should prioritize them as likely pathogenic variants. So we've seen this general trend of mutations decreasing nucleation at the C-terminus and mutations at the N-terminus increasing nucleation. But of course, there are always some exceptions that sometimes are a bit difficult to rationalize. And I will just give you an example. So there's this subset of mutations that I label here with a yellow dot. These are deletions that remove residues from both the N and the C-terminus, and they are strongly increasing nucleation. So it was a bit difficult for us to understand at the beginning why you would remove residues at the C-terminus and still nucleate. But when we checked their sequences, what we realized is that what is happening here is that these sequences are bringing an aliphatic stretch from the N-terminus close to the partially deleted C-terminus. And so we hypothesized that maybe what's going on here is that what they are rearranging is an alternative aliphatic core. But still, there were some other variants that were quite similar to these ones. They were also rearranging alternative aliphatic cores, and yet they were not able to nucleate. But in the end, what we found is that only those variants with an alternative core that had a very specific length of this core were the ones that were able to nucleate. And this length was the same or very similar to that of the wild-type core. So it's not only a matter of rearranging an aliphatic core, but maintaining a specific length. Just to summarize, I've shown you here our atlas of amyloid aggregation. This was the first systematic comparison of different mutation types in a human disease gene. This gave us insights of specific conformations that are relevant for amyloid nucleation. And we think that demutagenesis is a powerful tool to study disorder regions such as the N-terminus of A-beta. And in our atlas, we also discovered more than 300 likely pathogenic new variants that were of unknown clinical significance. And finally, if predicting the mutations was the impact of mutations on amyloid nucleation was already quite difficult at the level of single amino acid substitutions, this is even more difficult when it comes to other type of genetic variants, and we could run very few of these predictors and just with single amino acid insertions and deletions and not for large deletions. And they perform very poorly. So overall here, we think that we need to perform more diverse experimental data sets if we want to build a new generation of predictors. And just to finish, I want to thank again people in the lab, and thank you all for your attention.
Thank you, Dr. Mireia Seuma. Fantastic talk. So we can take any questions if anyone has any. You can unmute yourself and ask your question, or you can also type them into the chat and I can read them out loud.
I have a question. So you show that most of the one on the right, I guess that's the C terminus ones have not affected nucleation scores, but there are some exceptions, if I remember from your MAVE matrix. And I'm just wondering, with those exceptions are like the reds that are like in the ocean of blues, have you looked into those specific amino acid substitutions and what happened there?
You mean at the end terminus? At the level of
the unstructured side, where most of them don't change nucleation. There are some that seems to change a little bit. Some substitution, if you look at a heat map.
Right, let me go to the heat map. There you go. So you mean at the C terminus that we have some blue substitutions, right? So these ones would increase nucleation.
Yeah. So most of them don't increase, right? But then some increase and some are pathogenic. So do you know why those specific ones? And they're actually seen in, because if I don't mistaken, the outline ones are the ones seen in patients as well.
Right. So for example, this one is a pathogenic mutation. Exactly. So I think we see some enrichment of some specific mutations such as valine or isoleucine. These are changes from aliphatic to another aliphatic. So we think that this is just on how you can pack this amyloid core. So you would expect that changing an hydrophobic to an hydrophobic, it wouldn't make much of a change. But maybe since this is very tightly packed, maybe you can just don't make the same type of interaction, right? And also, if you see at the C terminus, there's the last position of the peptide, which is quite interesting, which is the alanine 42, and this is known to make some interaction with the second monomer, and so it's definitely doing something different from the rest of the course. So here, yes, we would expect this position to act a bit different from the rest of the C terminus. And there are some cases that definitely we would have never predicted, and in the end is this thing that you better do the experiment if you want to understand the impact of these mutations, right?
Very interesting. Thanks.
I have a quick question. Have you been able to compare patients maybe in like a biobank or anything, like compare patients that have variants in the C terminus versus N terminus and see if there's any differences in either disease occurrence or like even like age of onset or anything like that?
So we see that most mutations that are associated to disease, the ones that you see in this heat map in a black square, most of them are enriched at the N terminus. There are only 2 mutations at the C terminus that have been associated to disease. And we really think that the N terminus has a role in messing up the conformation of these fibrils, and maybe it just speeds up this nucleation and the formation of these fibrils. And it's also interesting because the C terminus has been very well studied by structural methods because it's very easy to solve these structures, but the N terminus has been more difficult because it's disordered in most of these structures, but we really think that that's the important part to look at because it's where most disease mutations happen. So there's definitely some bias between the 2 parts.
Thank you. And Amar has a question. Do you want to ask?
Right. It's for these heat maps of amyloid nucleation, have you done any, like specifically in like the hotspots, have you taken into account any like neighbor or pairwise interactions between the residues or done any modeling like that?
Not yet, mainly because of
Data.
because of the data, because of how this library was designed, but definitely this will be one of the next steps, because we really see that we really envision that there are combination of mutations that can rescue, especially at the C-terminus, right?
Yeah, yeah. I would be very excited to see that.
Right, me too.
Okay. I don't see anyone else has any more questions. Thank you so much, Dr. Seuma, for coming today and giving your talk.
Thanks for having me.